So hi class, good afternoon. Welcome to the fall 2021 class of molecular biology. Today is the first class of this semester, fall 2021. So I welcome you all and I hope that this will be very enriching to you and we will have a good time learning new things and so in this class, I'm mainly going to cover the syllabus. So, my name is, so brief introduction about myself. My name is Shiraz Mujtaba and I'm a PhD. I did my PhD now many years back, maybe 20 years back. And the area of my research during PhD was on HIV. And I looked at genetic heterogeneity and various immunological markers in AIDS patients. So while I was doing my PhD was mainly was with patients. So I used to work with HIV/AIDS patient. I used to collect their samples, blood, look for various immunological markers, look for various co-infections, isolate the virus to look at the genetic heterogeneity. Because viruses, they mutate. And this mutation in viruses can affect the development of vaccines. So that was the area when I was working on my PhD. When I finished my PhD, I wanted to continue my research on HIV and AIDS, but I was while I was working with patients, I was always used to wonder that why different patients show different kinds of symptoms, why they're so different from each other. So of course, genetics plays an important role in constructing our immune systems. Genetics plays a very important role, and we all have same DNA, but we could have different function, different personalities. So I was always, having been working for patients for 5-6 years, I wanted to look at the transcription. I wanted to look at DNA, how transcription in different patients happen, what kind of proteins, what are the level of proteins. So all these led to do more intense molecular biology research. So I got a postdoc position at Mount Sinai School of Medicine on Upper East Side. And there, while working in the as a postdoc, I discovered the mechanism of HIV replication at Mount Sinai and published in a very reputed journal. And that was the start of my journey in US when I came from India. And then with that time, there was a new field of epigenetics that was coming into biological area. So I was working I won't go into the details. So after working on HIV, I worked one of the most important proteins called as P53, tumor suppressor protein that controls cell cycle. And so I got into more deeper into epigenetics and how our genes are regulated in different cells. So then I got a faculty position at Mount Sinai itself. And I was at Mount Sinai for a long time, almost for 13-14 years. And in 2013-14, I moved to Medgar Evers College. And at Medgar Evers College, I have been teaching BIO461, BIO211, for which I'm also the course coordinator. And I teach in the springtime BIO472, that is molecular biotechnology. And also sometimes I teach a senior seminar. And in past, I've also taught Bio 201, 202, so many courses. So that's a brief introduction for me. Now, these classes are asynchronous. So of course, although I have put my office address, but we are meeting on Zoom, we are meeting virtually. So I will go one by one. So look at my office hours. Somebody was talking about office hours. So my Monday is the longest office hours I have, from 1 p.m. to 2:30 p.m. Then on Wednesday, I have 2 p.m. to 3 p.m., my office hours. And on Thursday also, 2 p.m. to 3 p.m. And below this, there is a Zoom link. This is a recurring Zoom link. I don't have to give you a Zoom link all the time. So I just posted the Zoom link on the Blackboard. And I believe that you were able to access, that's why you are here. So I'm a very, I try to be as much organized as possible, and I mainly work with, we have no choice in this virtual climate that we have to utilize Blackboard. So in Blackboard, let me show you how I organize my lecture. So in Blackboard, I'll just show you one of the classes that I have taught during summer. So, or let's go to the molecular biology class from the last semester, how I organize the class on the Blackboard. And I'll try to follow the same things. So if you see the content area, if you go to the content, so I go by learning activities. So we are meeting once in a week. So I will upload here learning activities for each week, right? So for each week, for example, there will be history of DNA. Let's say for the next week, there will be history of DNA. Then there will be recording for the chapter 1, then the assignment, and I will also post the recording for the assignment. And there could be sometimes a topic to discuss on the discussion board. So that's how my Blackboard, that's how I'm going to organize this semester also. In the Blackboard, in the content area, for example, today, I will create a content folder and I will say here, syllabus. Syllabus Bio 461, Fall 2021. And then I would upload the syllabus. So this is a folder, and so I created a folder. Within this folder, I will create an item, and this item would say syllabus, Bio 461, Fall 2021, and then I would upload this syllabus, what I'm talking to you right now. And here I will make accessible this syllabus to you. Let's say, most likely we will have the exams by mid-December, but I'll keep this syllabus open till the end of this year. So this is how I am going to organize my content. And so to go back again, you see here the syllabus, and you press the link, you get the syllabus here. And then later today, I will upload the recording because after I do this Zoom recording, I'll have to convert into YouTube. And then we go from there. So that's how my Blackboard will be organized. So now I give a lot of importance to syllabus, because syllabus is a guiding force that guides you and me. Because there is no limit to teaching or to learning, and these days everything is so much available. It is so vast, but for the efficiency, for maximization of learning, we have to follow some objectives. And these objectives are called as student learning objectives. And that is expected out of you to learn, and that is expected out of me to teach you. So if you go to, so on the first page, number 1 of the syllabus, there is my address, Zoom link, welcome message, and course overview. On page number 3, there is a student learning objectives. So there are 11 objectives for this syllabus, for this course. Number 1 is design experiments to prove that DNA is the genetic material. So there has been a lot of, knowledge grows, right? Knowledge evolves. It is not absolute, and and still we don't. It is hard to pinpoint that what came on this planet Earth first. Was it protein? Was it DNA? Was it RNA? You can argue from each point of view, but from the point of view of consensus, DNA has been chosen to be the genetic material because there can be various types of RNAs, there can be million types of proteins, but DNA is only of one type. So people from RNA biology, people who work on RNA, they think that RNA is more diverse, more functional, so that should be the genetic material. But somehow conventionally, we have chosen DNA to be the genetic material. So, so now scientists, they have done various kinds of experiments to prove or disprove that DNA is the genetic material. So in the chapter number 1, we will systematically cover to study how scientists came to the, or how molecular biologists, or how biologists came to conclusion that DNA is the genetic material. So that is the student objective number 1. The second SLO is compare the general organization of prokaryotes and eukaryotic genome. Now you can ask me a question that why we are studying bacteria, why aren't we going into eukaryotic cells or human cells? So my answer to this question of yours will be that there are many things of molecular biology and biology that we learn from bacteria. Bacteria can cause disease, but bacteria is one of the most important organism that can that has been studied by the scientists in the lab with regards to replication, transcription, so on and so forth. So first we are going to cover bacteria, and then we go towards eukaryotic or human cells. Then after learning that DNA is a genetic material, we study the difference between bacteria and a human genome. What are the similarity? What are the differences? Then we go into studying how the DNA replicates itself. Now, why DNA has to replicate? Our body, we start our journey as a single cell, and then we develop into an individual inside our mother's body, and that goes for many multicellular organisms. So in multicellular organism, there is growth, differentiation, development. In single cell organisms like bacteria or yeast or algae, cells may not be multicellular composition. They can be simple single cell, but this single cell also wants to divide. Now, a cell doesn't divide in vacuum. It doesn't divide as an empty bag. Whenever a cell divides, it has to carry the DNA. Because if the DNA is not there, then body cannot function. DNA has program, DNA has those instructions that a cell that is relayed from DNA to all over the cells and all over to the body of a multicellular organisms. So before a cell divides, whether it's a multicellular organism or a single cell, there has to be replication. the DNA has to duplicate. Now, any cell that has to survive has to generate resources. One of the resources is protein, ATP, lipids. So those resources, one of the ways one can develop those resources is through transcription and translation. So that we will study as a student SLO number 3. Then we determine the mechanism, how transcription and translation occurs. In other words, we will learn to know how genetic code came into existence, what is the value of genetic code. And then, when there is a normal path, right, like DNA replication or DNA transcribes into RNA as transcription, then sometimes DNA is susceptible to mutations. So DNA, like our English alphabets, there are in the English literature, there are 26 alphabets. In the dictionary of DNA, there is only 4 alphabets, A, T, G, C. And this can change. And when DNA changes, it can change RNA and then subsequently protein. And these changes has a lot of potential to give rise to various kinds of diseases. So we'll study mutation and so on and so forth. We will look at how the gene is regulated and how DNA can be manipulated to develop into, because application of molecular biology is biotechnology. So this is a very fundamental course for you all. And many of you, if you're going towards a PhD program or going to medical school, this is a very important course for you. So there are 11 student learning objectives that and you should be a master, or you should be having a well-controlled, a good understanding of all these student learning objectives. And in a way, that is also my job to do as best as possible to teach you the whole syllabus of BIO461, which is called as molecular biology. So now I go to the book. This is a very simple book which is available on Amazon.com. This is called as Essential of Cell Biology by Alberts, Hopkins, Johnson, Morgan, and all those. So, so I will be posting the slides, and these slides will be maximum from this book. Sometimes I use material from outside. If I get a good slides, if I get good slide from outside, from Google Image or from other book, then I incorporate those slides into my teaching. But most of my teaching material is from this book. So now going back to the syllabus. Now let's look at the various composition of this course. So there will be 3 tests. Test 1, Test 2, Test 3. Test 3 will be your final exam. Test 1 and Test 2, they are 15% each. Test 3, which is final exam, is 20%. Quizzes, there could be 3 to 4 quizzes. The total quiz will be of 15%. Then one of the most important thing, as good as final exam, is assignments. And so because I try to give assignments during the class, but in this asynchronous, it is not possible. So this goes to you as a homework. So there will be a deadline for every week. So there will be assignment every week, and there will be a deadline. If there is a late submission, then you lose point. Then the discussion board. Discussion board is very important for virtual learning because it helps me to interact with you. I cannot see out of I believe 32 students, only 13 students are attending this class. But I have no ways to determine whether every student is participating or every student is trying to answer my question. So in addition to assignments, discussion board is very important, and that is 15%. So the weightage of final exam and assignments are same, 20%. Discussion board is as good as quizzes or test 1 and test 2. So please don't ignore a discussion board, and it is very important. So the first chapter is history of DNA. In the first chapter, I will be teaching how the scientist came to a conclusion that DNA is a genetic material, what was the starting point. And if you remember, if you have taken genetics class, Mendel studied in the pea plants how the characters are moving from one generation to the next generation. So what are these characters that the word word that Mendel used, from like dominant character or recessive character? What are these characters? So these characters are in other words, is chromosomes. And if you go deep, they are genes. And what are the genes? Genes are the sequence of DNA. So we'll try to understand in chapter 1 why DNA has been designated as a genetic material, right? So that's chapter number 1. Chapter number 2 is DNA and chromosomes, where we look into the structure of DNA, and we look at the structure of chromosomes, how chromosomes are made up of, so on and so forth. So once we look at the genetic material is DNA, then we look at the structure of DNA, the structure of chromosomes, then we go into DNA replication, how DNA replicates, and what are the mechanisms. So most of these processes are replication or transcription. They are like teamwork, where various proteins they participate. Various proteins participate. So the third chapter will be DNA replication. Fourth chapter will be transcription. After I finish the 4 chapters, we will have test number 1. Test number 1 is after the chapter number 4, and this test will be multiple choice questions, and it will be offered to you on Blackboard. There will be approximately 45 to 50 questions. So after test 1, we go into chapter number 6, which is regulation of gene expression. And then we study mutations. We could have another quiz number 4 at this point of time. The second last chapter is cell cycle or carcinogenesis. And in the end, we talk about techniques of molecular biology. In total, we have around 10 chapters. And in 10 chapters, we move from basic information of DNA to how DNA functions during replication and transcription, regulation of gene expression, cell cycle. Then after we finish carcinogenesis, cell cycle, we have test 2. So test 2 is cumulative from chapter number 1 to chapter number 9. Then we finish the last chapter, which is techniques of molecular biology. And we have test 3 or final exam. Okay. And all of these exams are multiple choice question, 50 questions, so on and so forth. So now I would like to stop my recording and ask for your questions. So I stopped the recording, and do you have any question? I see one question in the chat. Yeah, somebody was talking on the phone. You have to be very, very, please, you have to mute yourself because, people are trying to focus. And if you keep your phone on, then it is not good. So is there any question about syllabus?
Yes, Professor. Could you go over about the format of the quiz and exams, please?
About the format? Sure.
Yes.
Multiple choice questions. format will be multiple choice questions. Let's say quizzes could be 20 or 30 questions, or maybe 15 questions, depends upon the size of the chapters. But test 1, test 2, test 3, test 3, which is final exam, there will be 50 questions. And all the questions will be from the slides, from the assignments, so nothing beyond that.
Professor,
Yes.
will the quizzes and the exam be timed, and will there be more than one attempt?
Yeah, 2 attempts.
And will it be timed as well?
Yeah, everything is timed. What do you mean by time?
Okay. Like, would it be timed like 45 minutes, an hour?
Yeah.
Like, how long would
So let's say if there are 30 questions, then there will be 40, it will be 45 minutes.
Okay, thank you.
Professor, quick question. How important is the text in relevance to the test? Will most of the questions be from a conglomerate of the information you give online and the book, the text itself?
Yeah, I follow the book.
Okay.
All the books are, all the slides are from the book. As I mentioned to you, all the slides are from the book, but sometimes I need to explain something better or differently. Then I use, Google Images or some other books, but the basic skeletal material is from this book, which is given in the syllabus.
Okay. I do notice that there's a 4th edition that's much cheaper. I'm not 100% sure if it's a difference in text and pages.
I haven't compared, so honestly, I can't give you the best answer. But I feel that maximum, it should cover the 4th edition. But I haven't compared the content myself.
So we need the book, correct?
Yeah, I mean, like being a student myself, when I was a student, I needed book for everything. So it's very hard for me to say, no, you don't need book, and you rely on my teaching. Sometimes my students, not all the students like my teaching, but you have to either go to the slides and then the books or back and forth. Any other question? So if there are no questions, then I would end this class and I will post the recording on the Blackboard.
Sorry, one more question. The syllabus is now posted. The syllabus is now posted on Blackboard?
I just posted it.
Awesome, okay, thank you.
Maybe 20 minutes back or half an hour back, something.
Okay.
And I'm going to post the recording also. But it takes time sometimes to convert into YouTube. So by late evening, I will post the recording also.
And I know this class is asynchronous, so how important is it? Do you keep track of who logs in when you lecture on Mondays, or is that like,
no,
you don't?
No, I can't.
Okay.
That's why it is asynchronous. I can't say that everybody attend my class or everybody should attend my class.
Okay, because I have a class that's required to attend during this time.
If I expect that, then, for example, I was also teaching one of the classes called as BIO461. That's a synchronous format. So everybody is supposed to attend the class. But here, no, you are not. And I cannot say that you have to attend my class. And the other way to answer a question is that my recordings, none of the students have complained until now. And if you don't understand anything in my recording, then you have office hours. I'll explain. Did I answer your question?
Yes, you have. Thank you so much.
Yeah. No, we'll have fun, no problem. Last year was was quite, I was nervous because this is a very important class, but I could manage it. And we manage well. I didn't have any complaints or anything. And not only this, I teach a BIO461 senior seminar. In summer, in summer is more intense, no issues. But I don't say that there will not be any issues. So if there is an issue, then I'm here office hours, and I respond to emails pretty promptly. Okay.
Thank you, Professor. Have
You're
a good
welcome.
one.
Have a good semester.
I have another question, just the last question.
Yes,
Every
ma'am.
Monday, 2 to 4, you will be lecturing regardless of anything and recorded, right?
I will be posting the recordings for the chapters on Sunday. Okay.
Okay.
And if you have any question, then I will address here.
Okay, thank you so much.
Because I am, if I say that I am teaching and I'm expecting all the students to attend my class, I will be wrong.
Got you. Thank you.
Yeah. Any other questions? If not, then have a good semester, have a good day, and see you.
It's nice to see you again, Professor. You have a good day.
You too. Bye. Who is this?
James
Professor.
Thomas. I took you last semester.
Oh, okay. Okay, then I'm finishing the class. Bye.
Thank you. Have a good one.
Thank you.
Professor.
Thank you, Professor. Have a
Welcome.
great day.
Bye. Bye. You too.